So I know that puts me quite in a position. I know that everyone's probably hungry. It's not my fault yet. So let's see. I hope I can convince you that we actually have quite some exciting new products in the line of biologics support for your pharmaceutical R&D research and development operations. I'm showing you this slide. This is depicting a number of visuals of therapeutic modalities which you encounter in research these days. And if you look at what ChemAxon is traditionally focusing on, you can see that actually, well, this is just one little box which we cover with our JChem technology, and this is all about small molecules. Now, you might argue there are a couple of things, such as peptides, for example, which for many years have been kind of classified as small molecules, and people have been using traditional cheminformatics tools in order to address structure and data management of these kind of molecule classes. But what about all the others? Antibodies, antibody-drug conjugates, a number of nanoparticle conjugates. All these, to some extent, have structural features which prevent you from, well, describing them with an accurate and very exact chemical structure, because they might have essentially a distribution. You have a product which is not homogeneous, but you have a product which has a variety of different chemical modifications, maybe with some ambiguous locants, for example, on the sequence. For all of those, we offer the Biomolecule Toolkit, essentially a similar tool as we offer JChem-based for small molecules. The Biomolecule Toolkit is there for handling your data management on the large molecule space. And if you look through the feature list, I think you'll soon realize that it's actually much more than just JChem-based. It is essentially an API which we provide on Java level or on REST service level, with which we can cover a number of aspects which are relevant on housekeeping a set of large molecules. The first thing is we are fully committed to support the kind of emerging standard for large molecule representation, which is HELM, essentially an effort of the pharmaceutical industry covered by the Pistoia Alliance in order to meet the need of collaboration, data representation of large molecule space. We have tools integrated into this toolkit which help you with the standardization and canonicalization of the corresponding representations. There's a database storage layer which lets you put things into a database. We have control over different types of business logics in case you want to apply this as a real full-fledged registration system, including a couple of validations which you can run in order to, say, deduplicate your content, in order to validate that additional data you need to describe your entities is actually proper, set, and standardized. And once you have pushed all of this into a database, of course, you would like to query some of that content. You would like to have specific subsets of the molecules you have in the database. And what we provide you is the ability to kind of search this content based on biological sequences, as well as based on chemical substructures. But also if you kind of are exclusively describing your content with attributes and values, also on the corresponding metadata for your molecules, or sometimes they are not even molecules. One very important aspect here with respect to looking at those organizations who typically kind of manage their content currently with cheminformatics or bioinformatics tools is that we provide conversion utilities. We are able to convert from sequence space, FASTA files, for example, right into our representation layer, as well as coming from structural mol files. We can also convert them and perceive sequence within these chemical structure files in order to kind of let you enrich, essentially annotate your chemical structure files with sequence information, for example. Now, you've probably heard some of this already in previous years here in the US annual meetings. I think what is really important to kind of take home for you today is that we have reached a stage with these developments which we think are mature enough in order to provide them as integrated backend packages into our forthcoming end user applications. One of the kind of striking news is that we started to integrate the Biomolecule Toolkit into Instant JChem. This project has started about 9 months ago, and now with the August release, we're happy to provide a corresponding plugin which lets Instant JChem also manage your biological data content the same way as you're used to using it for small molecule structures. There are a couple of other integrations which are happening and taking place, and some of them I'll mention over the course of my talk. Another exciting news, and Bahram has mentioned this in the morning, is that, well, the Biomolecule Toolkit now got company. Please meet Eddie. Eddie is our answer to the demand that once you have a kind of backend toolkit which deals with the representation on a computer-based representation level, what kind of user interface do you use in order to, well, input and define such structures? So we have created essentially a web-based tool which lets you work on essentially sequence level together with additional modifications, together with non-structure elements if necessary, in case you have glycosylations which you're just naming through G0, G1, G2, but you don't specify them with actual structures. If you have nanoparticles, you have your gold 10 nanometer particle. This is part of your structure, but in itself, it doesn't come with something which we would typically describe in a chemical structure space. You see here the views is switching back and forth between 2 different view configurations. The first one gives kind of the highlights on the actual chemical modifications, so it's a little more detailed than the second part, which kind of lets you look at the sequence of a larger protein, for example. And the protein is annotated with a number of, well, identifiers as you've downloaded them from UniProt, for example. What I'm going to show you next, I think I've mentioned those, so let's skip that, is a quick example where you see the bioeditor in action. So what we are trying to do first is we try to draw oxytocin right from scratch. For this, we are loading a sequence which we have copy-pasted somehow into our editor. I can load the sequence here. It's going to be displayed. And if you're familiar with what oxytocin is, essentially, it's a 9 amino acid residue peptide hormone with a disulfide bridge between the 2 cysteines and an amidation at the C-terminus. So we are going to drag in the amino group for the amidation at the glycine end, draw a bond, and a similar way, we're going to create a disulfide bridge between the 2 cysteines. Now it will ask you which atoms actually to connect, make sure that we're making the connection between the side chain atoms. And then I can grab the whole thing and export it in a variety of different formats. We're choosing Helm 2 here at the moment, and this would be your kind of SMIOS for a large molecule. Now we can do the same thing in reverse as well. So we use our name-to-structure technology within Marvin.js just in order to load oxytocin. Then we export this structure which we just created as a mol file, store in our computer, and then go back into our BioEddie, choose the mol file, and when we load, we actually get the same molecule back. Now, what I'm trying to kind of explain next are essentially some case studies, things, what you can do. The first one is looking at the Campbell Content. It's a database which since about 1.5 years is offering a number of their peptide molecules in Helm format as well. And we've used the ability to kind of grab content from this database, perform our own conversion into Helm format, and then compare it back with what they offer as their kind of Helm annotations, just in order to check kind of a benchmark, how good is our conversion. So they have around 20,000 peptides. The Campbell interface then lets you search by a compound ID, by chemical structure, or by select metadata. But for example, you wouldn't have the chance to ask for a specific subsequence. The first step is we grabbed all of the mol file from this 20,000 peptide set, and then went through basically a very standard workflow in order to standardize these structures. The tools we used for that is KNIME and our standardizer. We kind of did everything you can read on the left side, strip salt solvents, remove hydrogens, neutralize structure, so that we have a real clear-cut standardized chemical structure before we actually go into the conversion and say, now we do the perception of the individual building blocks within that structure. Interestingly, what we have found is that there are a couple of examples which have a side chain linkage to the glutamate, which is recognized in the Campbell content as a continuous regular backbone peptide sequence, whilst we clearly could distinguish the cases where these were translated wrongly in the Campbell content, and hence we could correct it in 3 of those cases. The next step is largely about making sure that the kind of representation you've created can be unique. And as a matter of fact, when we kind of compared our canonical notations with the ones provided in the Campbell set, around 10% of the given Helm notations were differing, which is no surprise because we do a number of reorders in order to get to a canonical Helm notation in this case. But what's more crucial is that we were identifying something like 40 duplicate structures based on our canonicalization, which in case you're looking for all of the data you have for a given compound, is essentially you are missing out on some of the content if you don't do that canonicalization. The last step is then just giving an indication for what types of additional questions you can ask into that content. So we can search by sequence, including wildcards in case you have like sequence domains or specific active patterns in your sequence. You can look for metadata, everything which has oxytocin somehow in its name field. But we can also ask specific questions such as, give me back all molecules which contain a specific chemical building block, which is provided either by name or by chemical structure, for example. The second use case I would like to introduce to you is something which I believe is quite important because it tells you how flexible the toolkit actually is in setting up how an entity can be represented. So we're working with a small biotech company in Belgium who have the need for registering antibodies in their systems. And they are running instant JChem with the Minireg system for a while for some of their small molecule compounds, and now they have kind of added into their repertoire a number of antibodies. But these antibodies aren't fully defined, so essentially what they're having is the variable sequence of the light chain, the variable sequence of the heavy chain, together with a name field which describes, okay, this is the backbone or scaffold for the antibody. And then they would like to kind of, from an Excel template spreadsheet, which they receive from the manufacturer delivering these samples to them, register these definitions, unique them, standardize them, and finally measure data against those and use instant JChem as a reporting tool against this data. So this is, I think, currently in a test run at their site, and they are seemingly happy with it. So I think it's just a demonstration of that you don't need a full-fledged information about like the full sequence of your antibody in order to use the tool for proper data management. What I'd like to convince you is that the Biomolecule Toolkit is essentially a backend platform which you can use in order to enrich a number of applications with the ability to perceive and represent large molecule space. On the left-hand side, essentially, you have the registration database, which captures all of the information in a centralized storage, which is then accessed via the Biomolecule Toolkit and its data access layer. And this can be integrated into a number of applications. Now, I've mentioned instant JChem. We also have a peptide and oligonucleotide registration-focused web client. We're planning to move this on into the Plexus suite space, so Plexus Connect will be extended with the abilities you're now able to find in instant JChem. And we certainly would like to also include JChem for Office over the course of 2017 in these kind of rollouts. Any other application, what you're interested in and you think has a lack in kind of structure representation for large biologics, come to us. We should talk about this. Now, a couple of examples, just that you see some kind of screenshots. So here, what we can do is we can create a new project within InstantJChem, which points to a database hosting a number of biologics. And then you can see, okay, we can create a grid view, which visualizes the molecules now with our new editor as the renderer behind. And additionally, it also fully supports the kind of form views with additional data, as well as querying on a couple of properties of these molecules. We have a collaboration with a Swiss company called Texelia. They are developing a data management and tracking tool for essentially the classic steps from molecular biology, creating primers, using those primers in order to create plasmids, transfecting cell lines or microbes, and then afterwards expressing proteins. They're using in the back end for most of the kind of molecular assets, store them away and register them in a database, the Biomolecule Toolkit by now. And we are also having IDBS here with us in the room, where we have a quite successful collaboration in the chemical space, but we would like to extend these abilities into the biologic space as well. So this is a proof of concept which we've done, where essentially instead of providing a new chemical structure into your experiment in your notebook, you are now able to kind of define a biological molecule using our new editor. And once this is done, you have a small button which lets you register this thing according to the setup configuration in the Biomolecule Toolkit server application. And then you can return back the ID of the registered compound. It literally works just as it would work for small molecule registration. And I think that's a pretty exciting thing. On our roadmap, I think a lot of this is repeating what I already mentioned. So we're working very hard in order to get this support into Plexus Connect. We have a number of things which are quite high ranking on our agenda, largely on the bulk modification and bulk molecule handling. So the editor as such right now is a kind of single molecule application if you want. If you really intend to kind of do bigger sets, then you either use InstantJChem or Plexus Connect, or a yet to be developed tool which kind of allows you to do like bulk editing of sequences, for example. That's especially important if you systematically work in oligonucleotide space, say, and you have a couple of modifications which you run each original sequence through. First half of next year, we're looking into trying to provide you with tools which give you decision support for further development steps, essentially looking at how we can improve your design processes for sets of large molecules, assessing developmentability, that's quite a mouthful, and looking into structure-activity relationship analysis as well. And then further out, not really specified over the course of 2017, will be a date when the integration of the BioToolkit with the IDBS eWorkbook is probably happening, and also looking into finishing off the integration with JChem for Os's applications. In summary, I hope that I could demonstrate to you that with the Biomolecule Toolkits and the corresponding editor, you have a tool at hand which lets you curate and standardize even existing content which you might have in kind of molecular formats, chemical molecular formats, or in sequence space, and the ability to kind of enrich this content with what is missing from their respective representation. We try to bring it down to an application which is largely reminiscent of structure management, what you are used to with JChem Base and Marvin Sketch. And we have, feel free to kind of check out the demo station out there, a purely JavaScript-based tool, which you can use in browser and web environments in order to edit large molecule space. With this, I'd like to thank you for your attention.
What is the connection of bridges to the protein data bank when there is a real structure of that biomolecule? I mean, are you envisioning making bridges or URLs to connect the dots between your editor to existing 3D structures?
There is at least two aspects to this. One of them is that as the Helm standard is something which isn't driven by ChemAxon alone. With the Helm project, we are in conversation with a number of kind of public data providers, PDB amongst them, trying to look for an opportunity to kind of enrich the representation, what you find in the PDB database, and be able to kind of pull it off in Helm format to make it truly interconnective. However, the other point is that Helm is not meant to represent three-dimensional structured data. So in that respect, Helm aims to be a chemical description of which atoms are bound to each other, not necessarily their conformation in 3D space. So at some point, I envision that our tools will be able to kind of pull in a PDB file, perceive the chemistry inside, and if you have the coordinates given, I think Anders will show in Marvin Live that we have a kind of 3D viewer also made for the web browser where you can at least interrogate, this is how the structure looks like.
So you mentioned oligonucleotides at one point, but there was no siRNA or RNA of any kind in your initial array of structures. What is your plan for that, or even mixtures of, say, mRNAs for registering things like that?
So if you're looking at siRNA right now,
Right.
then I didn't show the example for that, but it's kind of fully supported with the limitation that if you really want to kind of use tools for the design process for these kind of molecules, then we still have a lot of work ahead of us in order to support the process. As soon as you have the product, you can put it in our database and it becomes available. But I also should say that there are a number of tools there which can actually assist you in that process. So very often what people need most is the final point of entry where they say, okay, now I need to kind of register a set of molecule candidates, for example.
I have a question about the canonicalized version of the Helm representation. How far is the validation? Let's say we can envision a biomolecule pretty complicated, like a multi-chain, cyclization, linkage, various type of stuff. Have you had a chance to sort of validate this uniqueness to ensure that indeed it is truly canonicalized version? Once you have a chemical entity in place, there's only one and only one Helm representation for that molecule.
We have run a number of tests on this. So we have essentially written our own canonicalization routine behind that. And there are limitations, I should state that. But in a realistic scenario, what people are producing, I have not seen that our canonicalization would fail. There are a number of special cases where we're just not there yet, but we also don't think that we actually have to really work on these cases, which are essentially symmetric graphs. When you look at the Helm representation as a graph-based representation, in Smile's words, these occur quite frequently, but I've never seen anybody like using 4 peptide chains, linking them up in something which would be like a stereochemical conformation. So these were cases where we would fail at the moment, but again, I have never seen such kind of complexity or such structure in real-life use cases. If you're looking at antibodies, for example, you have these symmetric antibodies, maybe with different modifications on each chain. This is no problem at all for us. High-level symmetry with chirality, that's the point. Symmetry as such is working fine.
So you mentioned the IDBS integration with them using the Biomolecule Toolkit. Is the scope there, or is the impression that your recommendation, would it be that it seems like there's a lot of functionality in Instant JChem right now from that case study you were showing? Would it be your recommendation that, is that functionality based on the assumption that people would have sort of a standalone Biomolecule Toolkit registration independent, and that the IDBS e-notebook would sort of leverage that, or would that be sort of bundled together? Is the IDBS functionality dependent upon somebody having Biomolecule Toolkit sort of as a standalone entity for their registration?
I think this is something which we're yet discussing. as a matter of fact, I believe that as soon as you kind of have a substantial value of running the Biomolecule Toolkit as a standalone application, and you use it essentially independently of the e-workbook, then I believe this is how it should be the setup. There are two separate applications which you get separately from ChemAxon and from IDBS. There are a couple of functionalities which we also provide on the chemistry space. Essentially, like the drawing of a molecule, okay, that bundles Marvin Sketch, but it's nothing really special. So if we kind of bundle the bioeditor just in order to add a new entry, I think this should be covered under the license of the e-workbook. However, if you point out to a registration system and you have a separate user interface for administering and staging potential issues among the registered compounds, then I think you will require a separate application, and this is what you will get also in the case of the Biologics Toolkit.
I guess to clarify, so it seems like there's a lot that somebody could do currently right now with something like Instant JChem and the Biomolecule Toolkit, and I understood that the IDBS integration that adds sort of like the ability for the e-notebook to interpret sort of the Helm, so for it to work with the Helm standard, but that's almost seems like that's more of enabling the notebook rather than the user. I mean, there's plenty of things that could be done sort of independent of, that looked like that was 2017, that would be extended to them.
Yeah, that's correct. That's okay. What I would say is the kind of path to move forward. The toolkit is enabling. The toolkit is not just an application. It can be used inside other applications, or it can be used standalone in case you only need a server-side application, which does the things what I've mentioned before.